Good evening, ma'am.
Good evening, sir. Sir,
evening.
I'll make you host, sir. You can continue, sir.
Yes, ma'am. Yes. Thank you.
Yes, sir. Now you can continue, sir.
Yes, ma'am. Thank you. Good evening, my dear students.
Good evening, sir.
Good evening, Pa. Okay. Today, what we are going to discuss, the remaining part of the principles of inheritance. Okay. So last class, what we have discussed in our about there are so many symptoms. Okay. So many syndromes also we discuss, and today we are going to discuss the remaining portion. Okay. Particularly, you know very about glycolysis. Where it occurs, by glycolysis? Glycolysis takes place in the cytoplasm of the cell, isn't it? Okay. What is the meaning of glycolysis? The glucose molecules, a 6-carbon compound, already you know about. The 6-carbon compound undergoes the breakdown. As a result, you will get it 2,3-carbon compound is called as, what is that? Pyruvate, otherwise called as pyruvic acid. Okay. So the glucose molecules, when it converted into a pyruvic acid, during the conversions, how many changes occurs? There are 10 steps also occurs in glycolysis. When you are going to see the glycolysis, the glucose molecules converted into what is that? See that glucose molecule is there. So 6-carbon compound converted into pyruvate. This is a 3-carbon compound. So how many carbon compound you will get it? 2 pyruvate compounds you will get it. During this process, how many enzymes are involved? 10-step process is there. At the same time, 10 enzyme is also involved in this process. Okay. Glycolysis, note it down, my dear students. Where it occurs, glycolysis mostly in the cytoplasm of the cell. Okay, don't forget. After the conversion of pyruvate only, they enter into the citric acid cycle, otherwise called as TCA cycle, otherwise called as Krebs cycle. Already you know a little about, isn't it? So in this metabolism or degradation pathway, glucose molecules lyses to give a pyruvate molecule. So before this part, see here, the glucose converted into glucose 6-phosphate. Here the 6-phosphate is there, isn't it? The 6th position, the phosphate molecule is occupied. Okay, glucose molecule, this one. The 6th carbon atom contain phosphate molecule. That's why they have given as a name is called as glucose 6-phosphate. This phosphate comes from where, the ATP. Don't forget, my dear student, the ATP. The ATP, adenosine triphosphate is there. Listen here, adenosine triphosphate. It gives one phosphate molecule for this process, and it forms a adenosine diphosphate. The one phosphate molecules absorbed by joined with the glucose molecules. That's why which carbon atom? The 6th carbon atom. So that's why it's a 6th carbon atom occupy the phosphate. That's why it's named as a 6-phosphate. Glucose converted into glucose 6-phosphate with the help of the enzyme is called as hexokinase. Hexo means 6 carbon atom. Kinase means where there is a ATP molecules participation takes place. Okay, so otherwise called as energy recurring process. Note it down, my dear students. This is also same, also repeated here also. See there, this is also ATP. See here, phosphofructokinase. The kinase where there is a activity of the particular enzyme that plays the ATP recurring process occur. The hexokinase, here also the ATP is utilized. Okay, one phosphate molecule is used for what is that? Glucose into glucose 6-phosphate conversion. Likewise, here also see that the fructose 6-phosphate converted into fructose 1,6-bisphosphate. See, the first carbon atom is also occupy the phosphate molecules, and sixth place also occupy the phosphate molecule. That's why the 1,6-bisphosphate, bis means 2 phosphate molecules occupied. Here also the one ATP. Before this reaction, how many ATP molecules are utilized? Mean 2 ATP molecules are utilized. Okay, this is not a reason. Suppose if it is any one of the genes responsible for the production of hexokinase, phosphoglucoisomerase, and also phosphofructokinase. Likewise, imagine any one of the genes, suppose lack, what happen, you know, it will due to some disease. Likewise, the metabolic disorder occurs due to the gene or allele responsible for the production of particular enzyme. Okay, just like which one, you know, phenylketonuria. Phenylketonuria, phenylalanine converted into tyrosinase, isn't it? There is an enzyme is there. That enzyme secreted by the X-recessive gene. Likewise, here the glucose 6-phosphate dehydrogenase enzyme is lack. What happened? That things we are going to study the first one. See that one? See, the glucose 6-phosphate dehydrogenase deficiency. Okay, this is the another disorder. It is a genetic disorder. Note it down, my dear. It is a genetic disorder that occurs most often in males only. Sometimes they may ask the questions. Okay, the glucose 6-phosphate mostly affect male, female, none, or what is that? Any other option. Sometimes it may ask animal also, like that. The male is mostly affected in this disease due to the genetic disorder. This condition mainly affect red blood cells. Very, very important. Glucose is essential for the maintaining the structure of RBC molecules also, because it's carry the, what is that? Energy, ATP, from one place to another place also. It can transfer the one place to another place, and also, what is that? Distribute the ATP, it's energy currency. This condition mainly affect the red blood cells. So as a result, what happened? In affected individuals, a defect in enzymes called glucose 6-phosphate dehydrogenase. Which enzyme is the glucose 6-phosphate dehydrogenase? Dehydrogen means, what does that mean? The removal of hydrogen molecules. Okay, by the enzyme is called as dehydrogenase, causes red blood cells to break down prematurely. Before maturation, what happened? You know, the glucose, the RBC molecule completely denature or break down. As a result, it will not carry any, what is that? Substances. Okay, so this is very, very important. It is also one of the inherited means it's also comes from the parent. Okay, this condition is inherited in an X-linked recessive pattern. X-linked recessive pattern. Hold it down, my dear. So this is also one of the diseases. Here you must note it down. Glucose 6-phosphate dehydrogenase deficiency, right? Okay. Genetic disorder. Okay. Mostly affected males. Okay. What will be the condition? You know, mainly affect the red blood cells. Okay. Which enzyme responsible? Glucose 6-phosphate dehydrogenase. Okay, what will be the effect on it? The causes red blood cells to break down prematurely before maturation RBC. What happened? It can be easily destroyed. Okay. See that. This condition is inherited in an X-linked recessive pattern. Always, all the diseases. Sometimes it may be dominant character is the dominant gene is also responsible for the particular syndrome. But in this category, X-linked recessive pattern only. Okay. Don't forget, my dear. Go to the next one. See that one. This is the normal RBC cell. It's produced from where, the glycolysis takes place in the liver cells, in cytoplasm particularly. Okay. The blood glucose, but the blood glucose is here. The blood glucose converted into pyruvate, isn't it? Again, the pyruvate converted to lactate, otherwise called as lactate dehydrogenase. Okay. These are the conversion. Suppose if it is after the formation of pyruvate, they may be enter into the citric acid cycle. It's also another pathway. Okay. Okay. Another one, the pyruvate is also converted into which one, lactate in the which one, tissues, body cells. Okay. The blood lactate, again, it entered into the liver. So this is also a cyclic manner. Okay. What happened in the next one? See here. Glucose 6 phosphate deficiency disease. See here, the X-linked, already we told, causes a non-immune hemolytic anemia, infection, medication, fava beans. These are the cosmetic organisms, cosmetic agents. We may call this a cosmetic agents. Suppose infection due to the viral, bacterial, what happened now? Automatically RBC it will be lead to rupture. Suppose unwanted medicine, some of the medication, when you are taking the chemical substance, sometimes it may be rupture the RBC cells. And one more is the fava beans. It's also one of the beans already you know about. Okay. I will just go to the Google, we'll get the idea. Okay. These beans also sometimes what happen, lead to the hemolytic anemia. Okay. Then protects against malaria. Protects against the malaria is also. And Keynes bodies of smear, Combs test, negative, non-immunity. These are all the identifications. These are the glucose 6 phosphate deficiency. Sometimes may be reported in the which one, malarial case, but also Keynes bodies on smear. Smear means I have to take the blood sample in a single slide and take another slide, replace it and make it a smear. Okay. And this is the Combs negatives, non-immune mediated. These are the identification of the particular disease, the glucose 6 phosphate deficiency. See that glucose molecule is here. It converts into glucose 6 phosphate with the help of the enzyme is called as hexokinase. Here the ATP formation takes place. It is utilized here. The glucose 6 phosphate, what happened after converted into 6 phosphogluconate by the enzyme is called as glucose 6 phosphate dehydrogenase. Where there is a dehydrogenase enzymes involved, just imagine, don't forget. Here the NADP is also participate in this reaction. It's catalyzed the reactions. NADP converted into NADPH. Note it down, my dear. NADP is converted to NADPH. So nicotinic adenine dinucleotide phosphate hydrogenase. Okay, this is the thing. It is a no glutathione. These are all the simple things. Is that this is the equation. This is the equation. Suppose this is lack, means what happened? The conversion of glucose 6 phosphate into 6 phosphogluconate is not possible. So it is lead to some diseases. It will not carry any particular component from the one place to another place. As a result, stress, hemolytic anemia is also possible because it's rupture the RBC, isn't it? So it cannot, what is that? Carry oxygen molecules. So this is the a complete process of the glycolysis. Glucose 6 phosphate, fructose 6 phosphate, fructose 1,6 phosphate. Up to this level, these are all the glycolysis also divided into 2 parts. Okay, hexose 6 carbon path and also 3 carbon path. Up to this level only. Up to this level only. This level. Okay, fructose 6 phosphate. Fructose is also 6 carbon compound. So others are, see here, all are 3 carbon compound. So these are the 2 pathways. Glucose 6 phosphate, fructose 6-phosphate and fructose 1,6-phosphate up to this level. So there is 2 ATP molecules also involved. And after that, what happened? No, the fructose 1,6-bisphosphate converted to dihydroxyacetone phosphate glycerolidine phosphate. First, the glycerolidine 3-phosphate enter into the further reaction as a result of formation of 1 pyruvate molecules. Again, the dihydroxyacetone phosphate also converted into glycerolidine 3-phosphate. Again, it entered into the next pathway. As a result, you will get it 2 pyruvate. 1 glucose molecule, you will get 2 pyruvate molecules. Between these two, what are the ATP molecules? Also formation takes place here. 1 NADPH2, 1 ATP here, 1 ATP here. So 2 ATP molecules also takes place here. Production also takes place. 1 NADH2. Okay. 2 times repeated means how many? See, 1 NADH2, that is equal to 3 ATP molecule. Okay, 3. The fourth one, fifth one. So after this breakdown, how many ATP molecules synthesis takes place? 2 ATP. How many NADH2? Only 1. First, the glycerolidine 3-phosphate enter. Next, also the dihydroxyacetone also converted into some again glycerolidine 3-phosphate. Again, the glycerolidine 3-phosphate also enter into the conversion of pyruvate means. So the 2 cycles also repeated. Okay, so 1 plus 1, 2 NADPH2. That is equal to 6 ATP molecules. Here 2 ATP molecules also formation takes place. So second one is also 2. So 4 ATP, yeah, and 2 NADH2. Total 10 NADH2. 10 ATP molecule synthesis takes place in glycolysis. From this 10, the 2 molecules are utilized here. So total 8 ATP molecules. Don't forget, my dear. This is also sometimes they may ask the question in glycolysis. When you are going to study the respiration process, we will study that things. Okay, see that one. So next one is cystic fibrosis. Already we studied cystic fibrosis. It affects the respiratory and digestive system. Respiratory and digestive systems. Cystic fibrosis is a genetic disorder, isn't it? It is also one of the genetic disorder that affect the respiratory and digestive system. Okay. And people with the cystic fibrosis inherit a defective gene on chromosome 7. Cystic fibrosis, which gene is responsible, which chromosome means chromosome number 7. Don't forget. There is a production of abnormally thick, sticky. This is the very, very important thing. This thick, sticky mucus which clogs the airways in the lungs. Already we studied in previous slides also, isn't it? Thick mucus formation. The mucus, what happened, already you know about it, completely black the, which one, duct of the pancreas, so digestive system, and other respiratory system also. As a result, what happened? And also, it cannot get into the intestine. It will not enter into the intestine, particularly the digestive system, and mucus also black duct in the pancreas. Okay, so these are the products. Already we saw it, this one. Okay, cystic fibrosis. It black, the passage of the, what is that, air. As a result, what happened, it reduced the path, so we cannot able to breathe properly. Okay, this is lead to the genetic disorder, chromosome number 7. Okay, so go to the next one. See the cystic fibrosis, chromosome number 7. So this is the chromosomes. Here, chromosome 7 is the, see here, 7p22.3. This is nothing but the genes locations. This is what is that? Centromere. The upper part, the P. Note it down, my dear students. The chromosome, it has 2 arms connected with the centromere. The P arms is very short in nature. Don't forget, P arms very short in nature. Q arms is long in nature, but it contains number of genes, alleles, nucleotide. We can say genomes. Okay, so the chromosome 7 structure is this. So the upper part is P. The arms is very short in nature. The Q is the largest in nature. Okay, don't forget. And the next one is called as Hutchinson disease. What is by Hutchinson disease? Already we discussed. It is also one of the this is in a brain disorders that affect the person's ability to think, talk, and move. Those who are suffering from the Hutchinson disease, that particular person, what happened? No, they cannot be able to think properly. They cannot walk. They cannot move from. It is also caused by the mutation in a gene on the chromosome 4. First one, 7. Here, Hutchinson is a 4th one. This disease is inherited an autosomal dominant pattern. Don't forget, Hutchinson disease is a, which one you know? The autosomal dominant pattern. This is not a recessive pattern. This is a dominant pattern. Okay, 4. So letter dominant means 4th letter. The chromosome is also 4th one. Imagine Hutchinson. Okay, so it's mostly occurs in the brain. Okay, defect the brain itself. See that this is the structure. Hutchinson disease is the chromosome number 4th one. The mutated Hutchinson is also what happened incorporated with the some difficulties. 35 or more gag repeats. What is by gag? So this is called as what is that? Glutamic acid. The glutamic acid is repeated again and again and again. Number of copy is also duplicated. See that this portion. So up to this level. Okay, repeated. So it lead to some difficulties, neuron degeneration. As a result, what happened? No, the deposits now, the GAGs repeats. It is also deposited in the brain cells. As a result, what happened? You know, neuron degeneration. The cells are completely what happened? You know, not look like a normal one. It can be some difficulties. Okay, so as a result, it cannot carry the what is the signal properly. So there is a things. Okay. Next, Hutchin's muscular dystrophy. Muscular dystrophy. Note it down, my dear. Muscular means you know very about that very, very important things. The muscle contraction. There are 2 types of protein is very, very essential. What are they? Will you please? Dhanalakshmi. What are the 2 types of protein molecule present in the muscles?
Don't know, sir.
You don't know? Okay. What about Ritika?
Don't know, sir.
Okay. Anyhow, so muscles contain, you know, the if you want to contract, so then only you can do any work. You cannot fold your arms means we cannot lift anything. Okay. So they need a myosin. Okay. And also not only that, lactic acid is also very, very important. Lactate. And ATP also very, very important. So that's why the glucose molecule present in every cells. Okay. So as a result, suppose if it is any difficulties means what happened, you know, we cannot do our work properly. Rigor mortisms. Okay. Rigor mortism is also one of the lack of ATP molecule in the cells. Okay. Each and every cells. Suppose if you are glowing. What is the meaning? Suppose you are applying any one of the chemicals, any one of the cosmetics, you are glowing, isn't it? Suppose we are taking a green tea. What happened? You will become a young, look like a young. How? How it will be possible? Any idea, Dhanalakshmi? When you are drinking green tea, okay, what will be the effect inside your body?
So, it will result in weight loss, sir?
Weight loss is it's not express, you know. I mean, so it's cell, it is not depends upon the cell, you know, cell activities. It is not related to cell, isn't it? Cell rejuvenation. Okay, so otherwise called as antioxidant substance present in the green tea. Take an example of gloves. Okay, it also contains and also blackberry. They are all containing what is that? Antioxidant substance. Antioxidant. When you are consuming more amount of antioxidant, what happen? You know, your cell, it will not lead to old. It will not lead to old. It look like a young like structure. Young. So that's why your face or your skin also glooming. Okay, shining like that. Young in nature. Always in young. Okay. due to the consumption of these type of foods. So you must take antioxidant substance mostly present in the fruits, vegetables. Okay. So why I am telling, the muscular dystrophy means due to the some disease, chromosomal disorder, gene disorder, what happened, the muscular protein will be what happened? Defects. That is the reason. See here, the Dutchinson muscular dystrophy, the defect is cytoskeletal protein molecules called as dystrophin lead to impaired muscles functions. This is very, very important. Which protein is called as cytoskeletal protein present in the Dutchinson muscular dystrophy diseases means dystrophin. Dystrophin. Okay. Cytoskeletal protein dystrophin. Don't forget. Okay. Lead to the impaired muscle functions. Progressive weakness of the griddle muscles. Cardiomyopathy. Mental impairment. Respiratory failure in second or third decade. Okay, so these are the difficulties due to the chromosomal disorder. What happened with the particular protein molecules? What happened, it lead to the impaired muscles functions. It will not properly. Suppose the lack of this protein molecule, what happened? It will not properly. You cannot do any work like that. Progressive weakness of the griddle muscles. Progressive weakness. Weakness means, but the function is properly. Improper means what happened, As a result, the muscles also weak. Okay. Where it occurs, the cardiomyopathy. Heart is also contained cardio cells also affected by these protein molecules due to the genes or chromosomes. Mental impairment already in our report. Okay. And respiratory failure in second or third decade. These are all the symptoms of the Dutchinson muscular dystrophy. Okay, go to the next one. See that these are the structures. See, Dutchinson muscular dystrophy. So the mother is the normal carrier. See that the red color denoted as the normal. This is the affected one. The father is the normal. See that. So what will be the progeny? There is a 4 is possible. Is that? See. 2 years old affected persons completely affected, the 5-year-old normal, 8-year-old affected, 10-year-old normal, and then 15-year-old affected. These are the symptoms like this. Okay. So decades they told, that is the things. These are the difficulties. Okay. See that. So only male affected, but female may be carrier. Don't forget, my Dutchinson disease is also male affected only. Female it do not affect. Female only act as a carrier only. They can carry the molecule from the generation to generation. Okay. So this is very, very important one. See. And next one is called as alpha thalassemia. It is also one of the diseases, my dear students. Don't forget, alpha and beta thalassemia also present in it. The alpha thalassemia is a blood disorder. Note it down, my dear, blood disorder, because it is thalassemia. Thalassemia is denoted as a, which hemoglobin component. Don't forget. Alpha, beta, because hemoglobin contain how many polypeptide chain, there are 2 types of polypeptide chain is the 2 alpha chain, 2 beta chain. Suppose the difficulties in the alpha chain, that is considered as alpha thalassemia. Suppose if it is a beta, means beta thalassemia respectively. Okay, see that one. The alpha thalassemia is a blood disorder that reduces the production of hemoglobin. No, don't forget, my dear. Reduce the production of hemoglobin. Suppose lack of hemoglobin, what happened? They will not carry the oxygen molecule from the one place to another place or one tissue to another tissues. Okay. As a result, again, the same problem is there. Respiratory disorder and weakness. The carrier molecules also. What is that? Glucose and some other components they will not carry. So as a result, your body will be weak. Okay, see that. In people with the characteristic features of alpha thalassemia, a reduction in the amount of hemoglobin prevent enough oxygen from reaching the body tissues. So already told you, because hemoglobin, the main vital role is, what is that, carrying oxygen molecule from the one place to another place. Okay. Suppose the production of hemoglobin is less. So as a result, what happened, the carrying ability of also what may happen? Less. So as a result, it will lead to some disease. Okay, see here. And the beta thalassemia. So another one is called as beta thalassemia. It is also a blood disorder. It is also production of the which one reduce the production of hemoglobin. Both are same, but some variation is there compared to alpha as well as the beta thalassemia. It is classified into two types. Which one? Beta thalassemia is also classified into two types, depending one the severity of the symptom. According to the symptom only, the beta thalassemia is also divided into two types. One is called as thalassemia major, also known as Cooley's anemia. Don't forget, my dear. Sometimes they may ask the question. Cooley's anemia is also called as thalassemia major. Thalassemia major is also known as Cooley's anemia. Okay, this is where it occurs. The severity. Okay, severity of the symptom only. Second one is called as B, thalassemia intermedia. Okay, which one? The beta thalassemia also divided into two according to their severity of the symptoms. Okay, one is called as thalassemia major. The thalassemia major is also known as Cooley's anemia. And the B one is called as thalassemia intermedia. The Cooley's anemia is also called as, what is that? Thalassemia major. You can come to the conclusion like this. You can you can easily imagine like this. A, thalassemia major. Thalassemia major is called as Cooley's anemia. Thalassemia intermedia. Okay, these are the two types. Okay, of these two types, thalassemia major is a more severe. Of these two type of thalassemia, which one is severe one? The thalassemia major is severe one. That's why they have given us a Cooley's anemia. Okay, so this is symptoms. Many people with thalassemia major have a such severe symptom that they need frequent blood transfusion to replenish their red blood cells supply. Always what happens, you have to blood transfusion takes place. Then only they can survive. Otherwise, what happens? They will not survive. You have to repel. What is the replace it? Suppose the particular time duration, you have to replace the blood component or blood. So then only they can survive. Otherwise, they will not survive. That means it will lead to some diseases. It will take, suppose, a month, 6 months once or 2 months once. Okay, you have to blood transfusion. Which one? Thalassemia major. Such cases you have to do this one. Otherwise, it will lead to some difficulties. Okay, see that one. See, thalassemia disease, alpha and beta thalassemia. So this is a beta chain. Okay, which one is affected? Alpha chain is affected. That's why it's called alpha thalassemia. See here, the alpha beta chain is affected here. That's why it is called as beta thalassemia. I already told you, there are two types of polypeptide chain present in the hemoglobin molecule. 2 alpha chain and 2 beta chains. Suppose alpha means, what is that? Alpha affected. Alpha chain is affected. So beta chain is present. Beta means, what is that? Beta is affected. Alpha is present. Okay. So here the 4 oxygen molecules carry. Each and every hemoglobin molecule, 4 oxygen molecules carry out. But in this category, 2 is lack, means what happened now? The 2 only carries. So it is also what happened now? Lack of oxygen carrying capacity. Reduce the capacity of the oxygen transport from the one place to other tissues. Okay, these are the things. And then some genetic terms also given. Genetic terms also given in a book. This is also very, very important. Chromosome region also, 2 types is there. Already we discussed in the previous slides. The largest one and smallest one. In between there is a largest arm, smallest arm, like that, isn't it? Chromosome region P, and chromosome region Q. Gynandromorpho, note it down, my dear. Gynandromorpho. Morphologically, gynism as well as androgen present in it. Pseudogene. It is not a gene exactly similar to that of a human gene, but it is pseudo. Pseudo means false gene present in it. Hemizygous. Hemizygous, what is the meaning of the zygote present in the off only? We'll see one by one. These are the some genetic terms also given in your book, particularly what are the things, chromosome region B, chromosome region Q, and gynandromorpho, and pseudogene, and hemizygous. Okay, see that one. The chromosome gene P, a designation for a short arm of a chromosome. Don't forget, my dear. Which arm is very short? The chromosome region P. And so opposite of the P is Q. The chromosome region Q, a designation for the long arm of a chromosome is called as Q. So the P is the shortest one. Q, okay, is the longest one. Okay, see that one? You can easily identify. See that? This is the chromosome structures. There is a centromere is there. It's connected with the both arms. Already we discussed these things also. Okay. The upper part is the, see here, this is the smallest one, isn't it? The P is the smallest arm, this portion. This is the longest arms. This is the longest one, up to this level to the last one. This is the Q. Okay. So in between there is a band, is that in the each and every band there is a genome is also present in each and every genome. It contains what is that? Alleles. The alleles denoted your or our characteristics. Okay. So these are the very, very important things. And gynandromorpho. Morphologically, gynism as well as androgenism, both. Okay. Both characteristics present in a organism is called as gynandromorpho. Organisms that have both male and female cells and therefore express both male and female characteristics. Some of the animals, so many animals is that they are having the both male of female. Both the characters present in it. Okay. See the example you can easily identify it. So this see here. So these are the male and female. Example has given the off part only female, remaining of part only male. Likewise, I have selected a single slide and three examples. So these are all what is that? One side male, opposite side there is a female. Okay. So both the character present in it. That's why they have given as a name is called as gynandromorpho. Okay. Also male also having, isn't it? Male also having. See that one? This is also a roster. See that the blue color denoted as a which one, female, and that's one a male, like that. Okay, the both the character present. So this is also another one. So this spot is called as female. This spot, this is housefly, I think. Okay, and opposite side there is a what is a male. Both the character present the single organism, that is called as gynandromorpho. Morphologically, both the characters present in it. Okay, see this one. See that butterfly also, the off part will be female and remaining off part will be male. So that's also both. So these are the different types have been identified by the scientist. Okay. See that one. Pseudogenes. What is meant by pseudogenes? Already told you. It is non-functional gene is called as pseudogenes. The pseudogene gene, a sequence of DNA like a gene, but not functional probably. The remnant of the once function gene that accumulate mutations. Pseudogene, what happened, So it's similar to that of the gene, sequence of your DNA like a gene, but non-functional. They do not do any functional probably. Simply they present as it is. There is no any work. So such type, what is that? Called pseudogenes. Okay. See the next one. So these are the common defect of pseudogenes. Missing promoter. Suppose this is the structure. When you are going to study the lac operon concept. Okay, promoter and the operator. Okay, and inhibitor. Likewise, each and every one. Some of the gene also present in it, but there is no functions. Okay, here the missing promoter, missing starter codon, frame shift, premature stop codon, missing introns, partial deletion. These are all what is a common defect. These are the difficulties may occur. Okay, so this is the other one. And hemizygous, don't forget, my dear student, this is very, very important. Hemizygous. Okay, hemizygous means what does that mean? Having only one copy of a particular gene. For example, in humans, male or hemizygous because male gamete or male gene, male sex, what are the chromosome present in it? XX or XY? XY. Okay. What about the Y chromosome decide the male, isn't it?
Yes, sir.
Yes. Okay. So XY present in it, 50% of X is there and 50% of Y is there. Okay. So that's why hemi means what is that? Hemi or semi, like that. 50% of the female character is also present and 50% of the male is also present in it. So the male genes or chromosome is considered as a hemizygous. Okay, see that one, male chromosomes. See here. So this is the normal one. Homozygous alleles are same. Capital A, capital A. Here heterozygous. What is the meaning of heterozygous? Capital A and capital B and small b. This is the dominant alleles. This is also a dominant, but this is a recessive gene. But in this two, which one is that? Two different types of allele present in it. Capital B, small b is there. So such condition is called as heterozygous. So both are same, this is called as what is that? Homozygous. In this category, small a is also possible. It is also one of the homozygous, but recessive in nature. But in this category, capital A is the dominant in nature. See the last one, the C, there is a large chromosome is there, and another one is also there. This one is called as X chromosomes. This one is called as Y chromosome. In this part, 50% is Y, 50% is X. That's why the male gamete is called as what? Male alleles called as what is that? Only Y only present in it, isn't it? Only one Y only present in it. This is called as hemizygous. Compared to the others, female, there is both are XX. So it is called as what is that? Homozygous condition. Okay. Heterozygous means hemizygous is also one of the, what is that? Heterozygous. We cannot tell like that because heterozygous means, see that different genes also present, alleles are present in it. But in this category, one X chromosome is there, another one is Y is there. That's why equal proportion. Okay, it's only present in the male. That's why it is called as what is that? Hemizygous. Okay. During the gamete formation, how it will be divided, X and also Y. Okay. Take an example, female. Suppose the gamete formation is there. How it will be? X1. Another is also a X. Okay. So the male gamete is called as, or male gene is called as what is that? So chromosome is called as what is that? And hemizygous in nature. See that. And that's all about these lessons. We'll discuss some questions. Okay. Yesterday also one calculation is also there. So I will tell you one by one. You can easily identify. Which is correct about the Hutchinson disease? It is caused by the mutation gene on the chromosome 7. It is caused by the mutation in the gene on the chromosome 4. So, and also 14 and also 24. Which one is correct, my dear? You have to take the notes, then only it will be easy. So Hutchinson disease, chromosome number 4. Okay, don't forget, my dear. Chromosome number 4. Go to the next one. See that. In a chromosome, which arms is long? Which arms is long, ba? Which one is a long? P
Q
and
region.
Yeah, very good. P and Q is not there because both are present in it. P region is the upper part and Q region is the lower part. So the Q region is the longest one. It's a simple question only, directly asked. It is not exactly similar to that of the NEET question. It may be, sometimes it may be a match the following. I have asked 3 questions. These 3 questions also comes where? In match the following. Sometimes, which one the assess and reasoning. Okay, sometimes it may be a direct question also. Okay, so this is also very, very important. See that. Which of the following is called as Cooley's anemia? Iron deficiency, alpha thalassemia, beta thalassemia, and thalassemia major. Don't forget, my dear.
Thalassemia major.
Yes, okay. Thalassemia major is also called as Cooley's anemia. Okay, which genes? Hemizygous. Now only we studied, isn't it? Hemizygous means which one of the 4 the correct state? Which one is correct? Human female genes XX, human Turner syndrome, human male genes XY, and none. So have to tell the which one is correct one? XY, hemizygous. Okay, and then last one, gynoandromorpho. Which one is the correct statement? Okay, male characteristics only, both male and female characteristics, female characteristics only, none of this. Which one is one? Both gynoandromorpho, gynecium, andrecium. So both are present in it. That condition is called as what is that? Gynoandromorpho. Okay, both male and female characteristics present in it. That condition is called as what is that? Gynoandromorpho. Okay, so there are so many examples already we discussed today. So next one is what is that? So that's all about the principles of inheritance lesson completed. Next, another one is we will see the next classes. Okay, see here. Here you must note it down. Only one question. Yesterday we saw it, isn't it? So what is that? See here. That yesterday we saw it. One question I asked. Okay, the Hardy-Weinberg law, but I couldn't explain that times. So today we'll see the things. Okay, see that one. Can you see the question? Okay. Can you see the board bar?
Yes, sir.
Okay.
It's visible.
Okay. See the question here. The question is in a HW populations. In HW population, okay, 1,600 individual is there. Okay, and 256 are showing recessive triad. Don't forget, this is the main important. 256 are showing recessive triad. Find out that heterozygous individual. Okay, so here, which one is? We have to find out heterozygous condition. Heterozygous, what is the meaning of heterozygous? P and Q. See the formula here given. These are the options. These are the, see, these are the options given here. See here. Okay. So this is the question. So here we are going to find out the answer, how it will be. See this one. So this is the Hardy-Weinberg law, isn't it? What is the meaning of Hardy-Weinberg law? You know about that? Okay, see that? P squared plus Q squared, that is equal to 1. So given here, see here, P squared plus Q squared plus 2Q PQ, that is equal to 1. Isn't it? P squared we do not know. Q squared, that is called as recessive. This one is called as dominant. This one is called as dominant. This one is dominant. So here, given here, I think, okay, this one is dominant. This one is recessive. Okay, it is similar to that of the, which one, the molecular formula, A plus B whole square. Okay, when you are putting like this, what happened now? So A squared plus B squared plus 2AB, that is equal to 1. So this is the formula. The same things also repeated here, P squared and plus Q squared, that is equal to 2PQ, and that is equal to 1. So P squared plus Q squared plus 2PQ, that is equal to 1. This is Hardy-Weinberg law. Here, what they have been given here, 256 are showing the recessive. Okay, P is a dominant one, Q is a recessive one. That's why I have chosen here 256 into recessive character. Okay, that is what is the meaning? Q is a recessive, P is a dominant. Okay, and 2PQ you have to find out. This one we are going to find out because heterozygous condition. See here, the heterozygous means what is that? Both the characters present in it. Okay, the PQ. Okay, the formula, what is that? This is the Hardy-Weinberg law, already in our board. I have written total is what is that? 1600. The Q squared is what is that? Heterozygous conditions. Sorry, recessive condition is 256. You have to find out the recessive in percentage. Okay, 256. How many percentage? You have to calculate from the total. So 256 divided by 100 into 256 divided by 1600 into 100, that is equal to 16. Okay, what will happen? You will get Q squared is equal to 16. The Q is equal to 4. Okay, 4 how many percentage means divided by 100, you will get 0.4 percentage. Okay, 0.4 percentage. This is the P value. Q value, how will you calculate? So after that, get the Q value, you have to minus it. See that P squared as well as Q squared, that is equal to 1. You will get it. Which one? Q squared. How many percentage is the 0.4? Isn't it? 4 percentage is there. So if we find out the P squared, what happened? What have to do? What do you have to do? The 1 is here minus what is that? 0.4. How much you will get it? 0.6. Okay, so Q squared, already we have the answer. Okay, already we have the answer here, 0.4. When we will calculate the P squared, means the 1 minus Q, you will get P squared, isn't it? Okay, so P squared already we know about. P squared, that is equal to how much bar? Or P, that is equal to 0.4. What about Q? Sorry, Q is 0.4, P is 0.6, isn't it? Do you understand, my dear Kritika?
Yes, sir.
Okay, so here we have to find out the 2PQR, isn't it? This is the heterozygous condition. This is the heterozygous condition. We have to find out the answer, isn't it? So you have to substitute the value 2 into, see here, 0.6. The first one is called as what is that? P. P is what is that? 0.6. 0.6 into 0.4. So you will get which one? The 48. You will get 48. 2 into 6, 12. 12 into 4, 48. Okay, 0.48. The 48, what is that? 48 percentage. Okay, the 2 PQR percentage, how much is there now? In the total, 48%. The 48%, how much you are going to calculate? How will you calculate? 48% total already you know about. Okay, total already you know about 1600. See the another one, then only it will be better. Okay, see here, I will erase it. Okay, my dear. See here. See here. So 2 into, so you will get it 48, isn't it? 48%. What is that? 2 P Q, isn't it? 48%. So the answer is, what is that? You know, answer has given 748. How it will be coming? You know, there are 4 options has given there. Okay. Here, the question. So then only you can easily identify, no? So wait a minute, just on it. Yes. See that option is the 768 and 576, 133. Okay. You multiply these things, you know, 48%, how will you calculate, you know? See, 100%, listen here, 100% is which was how much, you know? 1600 means into 48%, how much? Okay, just strike out this 0 here, this isn't it? So you have to multiply into 16 into 48, 16 into 48. See here, so 16 into 48, 16 into 48. See here, 8 into 6, 48. So remaining 4 is the 188 plus 4, 12. Here 6 4s, 24. Remaining 2 is the 144 plus 642, isn't it? 4 plus 2, 6. So total answer is, see, 8 and then 6 and then 6 plus 1 is 7. The answer is, see here, the first one is the answer. So likewise, you have to calculate. Once again, repeat for you. I will finish it up to this level. Here you have to find out the which one, you know, heterozygous condition. Heterozygous condition means P is dominant. So both the P is present in. So in this category, you have to put P and P. So these both are dominant. Q is also a recessive. Q is a recessive genes. 2 PKR is the, what is that? Heterozygous condition. Here 256 has given as a recessive. So the Q has given, isn't it? You have to find out the first percentage. How many percentage of 256 out of 1600? So here we calculate the steps one by one. Total is 1600. Q squared is 256. This because it is a heterozygous. Okay. We have to find out the percentage means, see that 256 divided by 1600 into 100, you will get it 16. Q squared is 16 means Q is equal to 4. Okay. Converted into percentage means you will get it 0.4. Okay. Like 0. After getting the 0.4, what you have to do, you know, the equation there is a 1 is that because you will find out the Q value. So Q value minus from 1, you will get the P value. Okay. See here, find out the P2, the 1 minus 0.4, 1 minus 0.4, you will get 0.6. This is the Q value, isn't it? So now we can calculate it. P value we know, and Q value is also already we calculated. So you have to multiply with the 2PQ. Find out the 2PQ value. See here, 2 already you know what P value, and also the Q value is also there. So we'll get 48. This 48, how many percentage you will calculate means, see here, this is the 100 percentage, 1600 means 48 percentage, how much? So this is the result we will obtain. This is the correct answer. Okay. Do you have any doubt in this area, my dear? Dhanalakshmi? Ritika?
Sir?
No, sir.
No.
No, sir.
Just note it down this question and also type it in, check in the Google, you get the answer. Okay, there is a video is there. Yesterday I tried, but it will not possible. Today I calculate it. Okay, so that's all about this lesson part, completely finished, and we will pass on to the next one is molecular basis of inheritance. Okay, so see that one.
Thank you, sir.
Right, thank you. Take care. Bye.
Thank you, sir.
Thank you. Thank you, ma'am.
Thank you, sir.
Thank you, ma'am. Right.